Welcome everyone at the first tutorial of this summer school, and it will be about MaxQuant DDA. In the first part, we'll go through different experimental scenarios which you can encounter and proper parameters that you should set up for those experiments. The second part will be devoted to Andromeda configurations, like modifications and proteases configuration, partial processing, and Linux setup. Now, I will go slowly, but probably not as slow so you can follow my every move. On the USB sticks, there are assignments which copy everything I tell with screenshots. So I would recommend you focus now on the screen and what I'm doing right now, and later you can repeat everything in MaxQuant using those assignments. Also ask me questions on the go, but note that you can ask technical questions like where to find this or that parameter. For example, you didn't catch something that I was talking about during coffee, lunch breaks, and during Q&A sessions. So from that, I will move on to experimental scenarios. And the first scenario we'll be working on is most commonly used, in particular, label-free quantification, DDA. We'll use dataset acquired on the pull down of CDC23 protein from yeast. Here, yeast culture was lysed, and the subset of proteins was pulled down using antibodies. This subset was then digested with trypsin and forwarded to Orbitrap DDA mass analyzer. Also, the fragmentation was acquired on CID ion trap. So, moving to MaxQuant. When you open MaxQuant, first thing you see is the raw data tab. And the first thing you should do is to load your raw files. You can load your raw files with two options. First is load, where you can select separate files to load. And the second is load folder, where you select a folder and simultaneously load all the raw files inside this folder. Throughout the tutorial, I'll be using load folder option. So we press load folder and navigate to the folder with our raw data. In my case, it's assignment 1 MaxQuant DDA, same name as you have it on USB, and label-free proteome. We select the folder where all the raw files are contained and press OK. And once more OK. Here, now we'll see raw file names. If the file exists through the specified path, size of the file, and the data format. In this case, it's Thermo Fisher Orbitrap raw file. Next thing we should specify is experiment column. Experiment column defines how much files were acquired in different experimental conditions and how much quantification events you'll have in your output tables. Generally, all your biological and technical replicates you should specify as separate experiments. If you specify several files under one experiment, some intensities of those files will be summed. The only use case where you want to specify several files under one experiment is when you have a fractionated experiment. Now, this is not fractionated experiment, so we select one line and then press set experiment. And for this particular line, we want to name it P-histidine 301 as biological part and technical replicate part. Now, if you don't have fractions in your experiment design, you can also select all of the files and press No Fractions button. This will take differing parts of the file names and use it as experiment names. For example, here we have those parts differing, and this conveniently moves to experiment column. Lastly, you can specify experiments through Write Template option. When you press it, MaxQuant will write experiment design.txt template into the combined folder. We then go to the folder without data, go to the combined, and find experimental design template.txt. We can modify it in, for example, Excel very easily. And here we have columns like name, fraction, experiment, and PTM. Say we want to name our experiment the same as the name. Copy it here. I save the file. Close the Excel. And then I press read from file. Now I navigate to combined, find this experimental design template.txt, open it, and here I see that my experiment has changed to the names of the files. Those are all parameters that you need to specify for standard LFQ experiment. Note that there is also a parameter group column, which you can change. For example, select 3 files and then press set parameter group. One can change it to 1. It is useful when you want to apply different group-specific parameters to part of your raw files. This is not the case for this data, so I return it to the group 0. So now all of our raw files are in the same group. Moving on to group-specific parameters. First step in group-specific parameters is type. Here, standard refers to Orbitrap DDA, and we leave it as it is. And the modifications, default variable modifications are methionine oxidation and N-terminus acetylation, and fixed modifications is C-carbamidamethylation. In theory, one can specify any number of modifications from this table into this table, like so. However, with each modification you're looking for, the search space will increase significantly. So I wouldn't recommend adding more than 2 additional modifications into your search. In this experiment, we do not look for modifications, so we leave it as it is. Next is label-free quantification tab. We switch it from none to LFQ. Now you will see several LFQ parameters like LFQ mean ratio count that Carla mentioned yesterday, and usage of fast LFQ. Usually, again, you leave it as default, but for example, in cases where you want more reliable quantification, you can change LFQ minimum ratio count from 1 to 2. The digestion tab will have trypsin with proline sites as default, usually. If you want to add another protease, you, same as with modification, select something from the left table and add it to the right. We will not, because we only have trypsin in this experiment. Sometimes people want to use classical definition of trypsin. Then you will add this to the right side and remove this from the right side. Okay, I will get it back. Now, moving to global parameters. Are there any questions so far?
Yeah, I was just curious what you were referring to with the LFQ, changing it from 1 to 2, and if you could just expand on what that means.
So when LFQ calculates protein ratio, it looks for median of peptide ratios. You can, if you set it to 2, then protein groups that have less than 2 peptide ratios between samples will not be quantified between those samples. Now, by default, it's 1, and it allows us for much more data completeness. If there is any ratio between samples, it will be quantified with LFQ minimum ratio count 1. But if you take at least 2 ratios, then your quantification is more reliable.
Right. I was just curious here. There is an option for classic LFQ for single shots, but you recommend to choose fast LFQ, although the data is a single shot data. Is there a reason why fast LFQ is recommended over the classic LFQ, and what the difference would be?
Classic LFQ for single shots refers to LFQ algorithm that increases computational effort quadratically, right?
No.
So what is good about the classic LFQ algorithm is that it's scalable not only to single shot experiments, but also to fractions. And this is why LFQ algorithm has the part of this quadratic where we sum up quadratic ratios. And this mostly increases the quantification load for the MaxQuant. But when you have an experiment only with single shots, it's actually there is no much drop in quantification performance if you just use median normalization. So when MaxQuant essentially sees that you have only single shot experiments, it will just normalize by its median. When it sees you have fractions, it will normalize through classical LFQ normalization, but you can turn classical LFQ for single shots on as well. Yeah, you can do it as well. Fast LFQ refers to speeding up classical algorithm by constructing a graph between all samples and then selecting a subgraph from that sample that represents the whole distribution the most. That's what Carla was talking in his presentation, if you remember.
Thank you.
So for these experiments, you group 2 conditions, group 1, group 0. So I'm wondering how, like, you define like a group 0 or group 1 to make multiple groups?
So let's say, hypothetically, it's not about this experiment. This experiment was a single group. But let's say I set it to group 1, those 3 files. I then go to group-specific parameters, and I see here group 0 and group 1. And I move between them and make separate group-specific parameters for those groups. And here I use label-free quantification. In the second group, I don't use label-free quantifications. Here I press group 0. Here I pressed LFQ on. And now I pressed group 1. Here, LFQ none. By default, I can turn it on, turn it off, and make all kinds of separate group-specific parameters for group 1. So group 1 parameters will be applied to those 3 files, and group 0 parameters will be applied to those 3 files.
Is that the case if I have like a whole proteome together with phosphoproteome? Then I want to compare them.
That's actually a very good question. If when in the experiments, actually separating files by groups is the case for phosphoproteome, because in the phosphoproteome, you want to group all your proteome files in the first group and enable LFQ, and group all your phospho files in the second group and disable LFQ because it's enriched. It doesn't make sense to do LFQ.
Thank you.
I was just wondering, for the modifications, how do you pick the modifications that you'd put in for like the whole protein?
So on the table on the left, it's all the possible modifications. Now you select one modification and you press right arrow here, and you add it in the search.
So how do you decide? So for example, if you were to not, so for like a phosphoproteome, you'd pick the phospho.
Yeah.
But if you are searching for the total proteome, do people usually put in a modification?
No, because it increases the search space significantly. It's like you take your whole proteome and now you increase it twice as much for like one modification, 4 times for 2 modifications, and like gradually. Usually people, if they look for, in their experimental design, they wait that some modification will be increased. Then they look for that modification. Otherwise, it is unpractical to look for like every modification every time.
Okay, thank you.
Okay, I guess we should move on to global parameters now. In the global parameters, 2 main tabs are sequences and identification. In sequences, you will add your FASTA files. In this case, we have yeast organism. So we go to our FASTA file folder and look for FASTAs with yeast identifier. In this case, it's 559292. We select FASTA file and additional FASTA file, and we uploaded it. Now you see that you now have your FASTA file names. Organism is yeast, its taxonomy ID. Also, each FASTA file will have a description identifier rule, which is regular expression helping to identify which line is identifier, and description rule helping to identify which file is FASTA sequence. You can press identifier rule button and see different possibilities for those regular expressions. MaxQuant by default recognizes uniprot FASTA files, and it automatically picks the uniprot identifier. Same with description rule. Now, short section on how you download FASTA files. First off, you go to uniprot website. And select proteomes. And let's say we are interested in yeast. Here is the most used yeast proteome. We click on that. Click View Proteins, Uniprot KB. And then we can download our FASTA files through download. And we select canonical and isoforms. 2 files that I had, your canonical FASTA file and additional FASTA file canonical refers to Swiss proteins, and isoform refers to Tremble proteins. So you select this line and download it. Another way to download FASTA files is you look for UniProt entry that you're interested, copy it, and go to UniProt FTP website. So just Google UniProt FTP, go to uniprot.org FTP, current release, knowledge base, reference proteomes. For yeast, you go to eukaryota, and then you look for your UP entrance number. So you go there, and here you see FASTA file and additional FASTA file. This is the place where I took my FASTA files from. Now coming back to MaxQuant. This is all for sequences. And in identification tab, you will see a set of FDRs, ESM FDR, protein FDR, and you leave it as 1% unless you have a specific design of the experiments. For example, you have a peptidomic experiment, then you change protein FDR to 1. What we're interested right now here is match between runs. For standard LFQ experiments with shingle shots, you want to turn it on to increase identification performance. And here we will have a set of parameters defining retention time window, which is used to look for matches between runs. So you can also modify number of threads down here, and then you press start to start your search. This is all for our first data set. I'll just show it like this. Now, let's modify a bit our previous experimental scenario. Let's add another organism into our analysis. For example, this data set is also labeled free quantification, but human proteome is spiked with E. coli proteome in 4:1, 4:1 and 2:1 ratios. It is also acquired on Orbitrap DDA and digested with trypsin. Let's, by the way, if you want to remove your files, you select everything and press remove. Now let's load second folder. Which is multi-species proteome. Here, you can again set experiment with no fractions, but to make it more readable, I will put ratio names in the experiment names. So this is 4:1 sample, and this is 2:1. By the way, I'm moving. Sorry for moving faster. Is there questions about the global parameters?
So, quick question: what does the thread mean? The number of threads that you were saying 4, and default was 4. So
Number of threads.
Yeah.
It's number of cores you use for your analysis. By the way, if you're doing DDA experiments, rule of thumb is that you calculate number of threads. Each thread will take 2 GB of RAM memory. So say you have 8 GB on your laptop, then you take 4 threads for your analysis. Same, you have 16 threads, 16 GB, you take 8 threads, and so on and so on. For DDA, those numbers are higher. I think it's 4 GB per thread for Orbitrap DDA. Okay, so back to the multi-species proteome, we define experiments. In group-specific parameters, it's same standard type, no additional modifications, label free, trypsin digestion. And now in global parameters, let's remove yeast, FASTA, and we will add E. coli FASTA and human FASTA. You can see two separate organisms in the organism tab. In the identification tab right now, also we leave match between runs on, but we also check split protein groups by taxonomy ID. Now, in your output table, for each protein group row, you will have, so instead of one protein group result, you'll have it split by the taxonomy ID. So in one row, you'll have all the proteins from the human proteome, and another row will have all the proteins from the same protein group, but referred to the E. coli proteome.
So just a follow-up question. So if you do the metaproteomics or microbiome studies, should you check this box too, like some gut microbiome samples?
So for
Yes.
for metaproteomics analysis, this will only work if you are taking sequence from a reference database. So if you have your customized database, then you don't really have a taxonomic ID for each proteins.
So let's move then to the next data set. And the next data set is very different from the previous two because it's not a LFQ, it's a labeled data set. MS1 labeled, in particular, it's stable isotope labeling or SILAC. How this data set was acquired? Two sets of cells. First is control cells grown in light, unmodified media. The second set is stimulated with epithelial growth factor and cultivated in heavy media containing isotopes arginine 10 and lysine 8. Those samples are then mixed together and fractionated through isoelectric focusing. Here, protein in the pH gradient move under electric field until they reach their isoelectric focusing point. In total, proteins were separated into 24 fractions and digested with trypsin, and then analyzed with Orbitrap DDA. In this tutorial, we took the first two fractions of this experiment. Now, let's delete those files and load the third folder. Here. Because it is a fractionated experiment, and those two files are fractions, we specify same experiment for both of them. And now we specified that those are two fractions of the same experiment by selecting both of them and pressing set fractions. And the interval of fractions will be from 1 to 2 here. So here we have same experiment, first fraction, second fraction. Going to group-specific parameters, type. Again, standard, because this is Orbitrap DDA, but we change the multiplicity now from 1 to 2. Light channel will be unmodified here, so we do not select anything. But in heavy channel, we select arginine 10 and lysine 8. No additional modifications. We turn label-free quantification off because here we'll have only one experiment, and for label-free quantification, you need at least 2 experiments. In the global parameters, we will add human FASTA and we turn match between rounds on because as a rule of thumb, MaxQuant matches fractions with the same number or neighboring number, meaning -1, -1, +1. I remember there were a lot of questions yesterday about match between rounds and fractions. So if you have, for example, here only two fractions, then MaxQuant will match those two fractions. And this is because during fractionations, there is still some overlap. No matter what's the type of fractionation, there will be some overlap between those neighboring fractions, but not far away fractions. If we say would have one fractionated experiment and then the second fractionated experiment, then MaxQuant will match first fraction of first experiment to first fraction of the second experiment. Now, this is all for SILEC data. Any questions? Okay. And now our fourth data set is different in the sense that it was acquired on a different mass spectrometer than Orbitrap DDA. So this is again LFQ, kiloproteome, but acquired on TimsTOF DDA. TimsTOF mass spectrometer has two distinctive features. First is TIMS, or trapped ion mobility spectrometry, where ions drift through the tube under electric field filled with inert gas. And ions then are separated by their size and shape, which is reflected in physical property of CCS. They later go through time of flight mass spectrometer, where MZ values of ions are dependent on the time of flight before the detector. To load TimsTOF files, one cannot use load option, but only load folder option. This is because TimsTOF files are essentially folders. Here we see .d format, and if you press on it, it is folder with a lot of files. So now to load our TimsTOF files, we press load folder, find our data folder. Now you can press on any of the files and load them separately, or you can load the whole folder with TimsTOF files. Here you see that the data format has changed. Let's pass the specify experiments with no fractions. And in the group-specific parameters, when you press type, you will notice that MaxQuant automatically identified that this is TimsTOF files. And by default, he sets it to TimsTOF DDA. When you do TimsTOF experiments, you should always check that the type of your experiment is right here. Yes, this is all the other parameters are the same as for standard label-free quantification experiment. You turn LFQ on, you provide appropriate FASTA file, and you leave match between rounds on. Now, our last data set demonstrates another type of ion separation, but now it's not about going through the, it's not separated by CCS values, but rather their ion behavior in the changing electric field. And this technique is called asymmetric ion mobility spectrometry, high field asymmetric ion mobility spectrometry, or FAIMS. Usually, FAIMS is coupled with Orbitrap analyzer. And here, in this experiment, the compensation voltages are ranging between -25 and -70 voltages. So again, in TimsTOF, we have a constant electric field, and ions are separated by drifting through inerting gas. Here, electric field is changing from -25 to -70, and this way, ions are separated and supplied into Orbitrap DDA. So to load those files, you can use either load or load folder because they're essentially same Orbitrap files. Load this fifth folder. Specify experiments. And in the group-specific parameters, in the type section, we select FAIMS. Because this is not a labeled experiment, we do not select MS2 or MS3. We select regular FAIMS here. And all the other parameters are the same as for, actually, no. Actually, not all the parameters are the same as for LFQ experiment. In the global parameters, you turn match between runs off because it's just not implemented yet in the MaxQuant, the match between runs for FAIMS. So for now, keep it as off. Probably in the next releases, we will tell when you can use match between runs with FAIMS.